everybody, and welcome to the Chemistry 121 Supplemental Instruction series of videos. I'm Joey Smokey, and in this episode I'm going to be talking to you guys a whole bunch about the movement of particles in solutions. So there's going to be quite a few terms and vocabulary and all that kind of stuff, so you might want to be taking some notes as you listen along. Alright, so the first thing is diffusion, which is essentially the movement of particles. So if you have solutes in solution, like salt and water or whatever, those particles, the salt is going to be moving around in the water. And depending on how it moves, from one spot to another, is diffusion. Basically, it's moving around. Now, a concentration gradient is a difference in concentration. So let's say that at one part of the salt water there's a whole bunch of salt, but in another section of the salt water there's not as much salt. So what's going to happen in order to reach equilibrium, because everything in nature wants to be equal, kind of balanced out, the salt particles are going to diffuse from areas of high concentration to areas of low concentration. Basically they just kind of float along until the solution is even throughout. There's an even concentration. So diffusion is the movement of stuff from areas of high concentration to areas of low concentration, or you could also call this the diffusion down a concentration gradient. Now think about a beaker that you might have in lab, and you have a concentration of something in there. And then think about a membrane, like something you can stick between it to kind of separate two different sides of that beaker. So what that membrane is we can call it a semi-permeable membrane, meaning that it can basically separate two different concentrations. So maybe in one half of the beaker you have a high concentration of salt, and the other half you have a low concentration, and it's separated by this semi-permeable membrane. Now what the semi-permeable membrane does is it's selective to what it lets through. So for example, the salt particles, the little particles in there, can flow from the areas of high concentration to the areas of low concentration across the membrane, but the membrane won't let other things through, like maybe bigger particles that might be in the solution or water. It won't let those go through. So colloids are big particles. Basically, they're bigger things like enzymes or proteins or whatever. And there's also suspensions, which are really big particles, which you can sometimes see with the naked eye. So things like that, colloids and suspension particles, those won't go through a semi-permeable membrane because it's only selective to the smaller particles, like a salt particle. So the next thing we're going to talk about is osmosis. Now osmosis is the diffusion of water. Basically it's the movement of water between things. Now when you think about it, think about a highly concentrated solution. You have a whole bunch of particles in there, and there's not very much space between them. So in other words, there's not very much water between the particles. Now think about a lightly concentrated solution, a low concentration. You have a whole bunch of water between the particles. That's what makes it a low solution. Just like in diffusion, the water is going to want to kind of evenly distribute itself between all the particles. So it's going to move from areas of low concentration, where there's lots of water, to areas of high concentration, where there's not so much water. So that's what osmosis does. It moves from areas of low concentration to high concentration to kind of reach equilibrium. Okay? In other words, it's going to be going up the concentration gradient from low concentration to high. So now we're going to talk about three more terms. Tonicity. Isotonic basically means that two solutions are equal in their concentration. They're the same. Iso means the same. Hypotonic means that this particular solution, if it's hypotonic, it has less stuff than another solution. Hypertonic has more stuff than the other solution, so you can kind of think of it as the relative amounts of stuff in there. So what you guys as nurses, something that's going to be pretty important to you, is red blood cells and what you can do with them. So let's say that you have a red blood cell and it has a certain salt concentration, and you put the red blood cell in a hypotonic solution. What that means is that there is not as much salt in the solution, but there is more salt in the red blood cell. So the cell is hypertonic, it has more stuff, than the solution, which is hypotonic. So now think about osmosis. Where is the water going? Since the solution has more water per amount of stuff, the water is going to flow into the cell to reach equilibrium, to put more space between the salt inside the cell. So since the water is going inside the cell swells and it's eventually going to burst. That burst is called lysis. Now think about another example. If you put the red blood cell into a hypertonic solution. So hypertonic means that you have more salt in the solution and less salt in the red blood cell. So the red blood cell is going to be hypotonic while the solution is hypertonic. Think about the flow of water again. Where is the water going? Since there's more water inside the cell between particles than there is outside, the water is going to leave the cell. So eventually the cell is going to shrink and it's going to crenate. So basically it's like shriveling up. So the last thing to talk about is dialysis, which is basically selective diffusion. It can kind of go back to the semi-permeable membrane idea, that dialysis is going to only let certain things through between the membrane. It's basically just selective diffusion. If maybe you're only interested in letting one thing pass through the membrane, whereas you're keeping other things kind of separated, that's what dialysis is going to do. So there you go, a whole bunch of different stuff having to do with osmosis and diffusion, terminology, all that sort of stuff. Just basically understand where things are going, like the direction, down a concentration gradient, up a concentration gradient, wherever things are going to be going. That's all you got to worry about. So there you go. Go out there and have some fun with red blood cells.